Hello everyone, my name is Malcolm Burns. I'm the Principal Scientist for Food Authenticity Testing at LGC. I'm going to go through the introduction to the e-seminar today on the application of real-time PCR for food authenticity testing, inclusive of the quantitation of equine DNA. Before we start, I'd like to go over some important pieces of information. The following information pertains to a joint Defra Government Chemists Knowledge Transfer event that was delivered at LGC on the 17th of September 2015. Although every effort was made to ensure the information was correct at the time of delivery of the original knowledge transfer event, LGC and Defra assume no liability whatsoever on the accuracy of the enclosed information. LGC and Defra retain all copyright associated with the following e-seminar, inclusive of use of the audio recording and visual images. All rights are reserved. You may not distribute or commercially exploit the content. You may not transmit the content or store it in any other website or other form of electronic retrieval system without the express written permission of LGC and Defra. The aims of this e-seminar is really to welcome you and thank you for joining in on this knowledge transfer recording. The e-seminar aim itself was to provide access to information presented at the original joint Defra Government Chemist Knowledge Transfer event, which was held at LGC on the 17th of September 2015, which was looking at the application of real-time PCR for food authenticity testing, inclusive of the quantitation of equine DNA. This was a dual knowledge transfer event focusing on two separate aspects, the first of which was the application of the LGC or Defra draft SOP for quantitation of horse DNA, but also looking at the application of PCR and real-time PCR in general for food authenticity testing. The objectives were to disseminate the scope, purpose and application of the draft SOP for quantitation of horse DNA, inclusive of an introduction to the general principles contained within it, and also to provide examples of data analysis, and finally to provide guidance on the availability of methods and reference materials for food authenticity testing. We're now going to go through a recording on a presentation regarding the draft SOP for the quantitation of horse DNA relative to mammalian DNA in raw meat samples and provide some background to the SOP. As an overview to this presentation, we'll talk a little bit about the SOP for the quantitation of horse DNA, followed by the background behind the method, including its scope and purpose, application, the principle behind the method, some of the performance characteristics associated with this approach, the procedure before summarising. The SOP for quantitation of horse DNA is now available from Defra. And as background behind this, the SOP was developed through two previous Defra projects, FA0135 on the real-time PCR approach for quantitation of horse DNA, and the linked project FA0146 on method validation of the real-time PCR approach for the quantitation of horse DNA. These final project reports and the associated draft SOP have been approved by Defra. A follow-on work is ongoing at the current stage, involving an FSA collaborative trial of the real-time PCR approach for the quantitation of horse DNA, and a paper has been published in Analytical Methods describing the approach. EU guidance regarding meat speciation and meat quantitation has been published a couple of years ago. This involved some work by the EURL for animal proteins and feeding stuffs, which provided recommendations for detection of horse DNA, expressing the amount of horse meat in relation to other meat species on a weight per weight basis using a DNA approach, and also provided guidance on how to implement and test a threshold level of 1% weight per weight meat in meat. And there was a Commission recommendation on the 27th of March 2014 regarding a second coordinated control plan for horse adulteration in beef products. So Defra project FA0135 was the development of a real-time PCR approach for the quantitation of horse DNA. This revolved around the EU horse meat issue in 2013, where it was apparent there was a lack of guidance and scientific standardisation on how the amount of meat adulteration in a sample was expressed. This highlighted the requirement for a quantitative approach to be developed to accurately measure the total amount of horse DNA present in a sample. So the aim of this project was to develop a real-time PCR approach for the quantitation of horse DNA, and in line with current scientific thinking and showing synergy with the EU guidance on a similar approach, 2 nuclear DNA targets were chosen, one target specific for equine DNA and the other as a general reference target for any mammalian in a sample. The DNA target choice for use in the quantitation of horse meat in food samples using real-time PCR were based on single copy nuclear DNA targets suitable for quantitative analysis, for example the equine growth hormone gene, and we avoided using multiple copy mitochondrial DNA targets, which, although potentially useful for sensitive detection, may not be as useful for quantitative purposes. So regarding target identification, we had a look at candidate single copy nuclear DNA targets which could afford the potential for quantitation. This involved looking at the horse growth hormone receptor gene by Koppel et al. 2011. This was justified on the basis of high levels of assay specificity predicted for the target in silico, and also looking at mammalian and poultry myostatin gene by Lau et al. 2003, which was justified on the basis of high levels of sequence conservation existing. So we also provided meat samples as part of this project research and development, which were authenticated for species identity using a combination of real-time PCR, ELISA and DNA sequencing. And these were authenticated, and the authenticated meat samples were used to produce a range of weight per weight tissue gravimetric materials of raw lean horse meat in a background of raw lean beef meat on a gravimetric weight per weight basis. And the quantitative approach for horse DNA was validated in terms of a number of performance characteristics, including its specificity, the PCR efficiency and linearity associated with the approach, limits of detection, or LOD, as well as accuracy inclusive of trueness and precision. And the method was applied and tested on raw lean meat samples. Regarding the purpose and scope of the method itself uses real-time PCR to quantitate the amount of amplifiable horse DNA relative to the total amount of amplifiable mammalian DNA present in a sample. This method has been developed and validated for use with DNA extracted from samples that consist of raw lean horse meat in a raw lean beef meat background. However, the results can be equated to the equivalent amount of raw horse meat in a raw beef meat background on a gravimetric weight per weight basis. Again, to put this in context, the method can be applied to measure the amount of amplifiable horse DNA present in a range of samples. However, the method has not been validated for measuring the amount of horse meat relative to the total amount of meat in all cooked and processed samples. Now, due to the effects of food processing and cooking, the relative amount of horse DNA to total mammalian DNA may not be representative of the relative amount of horse meat in a sample for all cooked and processed foods and matrices. So the principle of the method: samples are analysed using a relative quantitation based approach, and the method uses 2 singleplex qPCR assays referred to earlier, one which targets a horse growth hormone receptor and one which targets mammalian and poultry myostatin gene. Calibration curves are generated from DNA extracted from 100% horse raw lean meat sample that's been treated identically to any test samples. These calibration curves are based on estimated genome equivalents. The test sample or samples themselves are then evaluated using both the horse-specific and the universal mammalian qPCR assays. Genome equivalent copy numbers for the test samples are then estimated and determined from the calibration curves. And finally, the percentage horse DNA content present is expressed as a ratio of horse genome equivalents relative to total mammalian genome equivalents. And anyone who is reasonably familiar with some of the GMO approaches, this is a very similar approach, for example, expressing the number of transgene copies of the GM relative to the total number of endogenous copies of the reference gene. So we've got a diagram here which basically shows the DNA being extracted from the 100% horse sample, which is then diluted and run on a appropriate real-time PCR machine to produce a calibration curve both for the horse specific assay shown on the bottom left and also a mammalian reference assay, producing good quality PCR metrics including linearity and R-squared. And when it comes to evaluating the test samples, which are gravimetrically prepared raw lean horse meat materials in a background of raw lean beef materials, these are treated exactly the same as the calibration curve in terms of DNA extraction. These are then run on the real-time PCR machine and challenged in terms of the horse assay and mammalian reference assay. CT or CQ values are produced on these, and these can be read off or extrapolated from the relevant calibration curves. Here we have some pictures of some of the calibration curves themselves and the amplification plots which are instrument specific both for the horse assay and mammalian assay, generating good calibration curves and good amplification plots as shown on the left and right. Regarding method performance characteristics, which were evaluated through the single lab study, specificity was looked at, and the Koppel horse-specific assay specificity was confirmed experimentally, showing no cross-reactivity with common meat species including beef, pork, lamb, duck, mouse, human and chicken DNA. However, the test, because it is equine, does cross-react with donkey. The Lau general mammalian and poultry assay targets the myostatin gene, and the DNA sequence is highly conserved between mammals, poultry and fish species. The absolute limit of detection was defined as the lowest target analyte concentration that could be detected on 95% of occasions. So the LOD for both assays, that is the horse assay and the mammalian assay, was determined experimentally to be less than 5 copies for raw meat samples. And the relative limit of quantitation, or LOQ, was defined as the lowest relative amount of horse content that could be reliably quantified using this assay, and the LOQ was determined experimentally as less than 0.1% for raw lean horse meat in a raw lean beef meat background. We've got some example results here which can also be found in the Defra final project report, showing the good comparability of the gravimetric materials and the expected values from the real-time PCR calibration curves using this approach, and showing good accuracy and good in terms of precision and trueness at the 0.1%, 0.5, 1, 5, 30% and 100% horse gravimetric weight-per-weight materials. So the real-time PCR approach has demonstrated a good accuracy in estimating levels of horse present in beef samples across a range of concentration. Here we've got a table that illustrates the example of horse and universal mammalian poultry qPCR assay performance data. It's based on 3 replicate plates using a 7.0 dilution series for the calibration curve, giving a dynamic range of roughly 20,500 down to 5 horse genome equivalents per target, and based on a triplicate PCR technical level of replication. Again, the mean R squared or correlation coefficient, the Pearson product moment correlation coefficient, is always above 0.99 there, and the slope of the line, which is indicative of the PCR efficiency, shows that the PCR efficiency is always above 90% for both assays. The procedure itself, as detailed in the SOP, provides information on the actual sequences in terms of the primers and the probes, and a single validated DNA extraction methodology should also be applied to the authenticated meat reference material, the horse, which is used as the calibrant, and all of the test samples. The calibrant itself consists of DNA derived from authenticated horse material, lean raw muscle tissue trimmed free of surface intermuscular fat and connective tissue, and the test materials should be quantitated, and the DNA extracts should be provided with appropriate quality metrics. We've got an example of an experimental design here on a 96 well plate. The top half of the plate is, for example, one of the assays, for example, Lao, and the bottom half, coloured green, could be the alternative assay, the Koppel qPCR assay. We've got a minimum triplicate PCR level, PCR technical replication level there for all standards, test samples, etc. We've got a horse DNA 7-point dilution series to be used as a calibrant, noted as S1 to S7, a PCR no template control as an NTC, extraction control, and a positive control, which is 50 nanograms of horse DNA, are put on the design to ensure data validity. And test samples are also provided as U1 to U6, again at a triplicate PCR level of replication. Regarding following on with the procedure, I've got a table here which is taken directly from the SOP, which describes some of the reagents to be used for the real-time PCR assay. The method was developed for a total volume of 25 microliters for the Lao mammalian reaction and 25 microliters for the Koppel horse reaction. Essentially, we combined 12.5 microliters of 2 times Techman Universal PCR master mix with the appropriate oligonucleotides, that being the forward primer, reverse primer, and probe, which are appropriate to the final reaction mixture concentration, and make this up to 20 microliters with DNAs or DNA-free water. And finally, add 5 microliters of our DNA template to make the total reaction volume up to 25 microliters. Again, here we have another table which is taken from the SOP illustrating the standards that are used, S1 to S7 for the calibration curve, and this is essentially based on preparing a 7-point or a 4-fold dilution series, S1 to S7, ranging from approximately 20,500 horse to 5 genome equivalent copies, based on 100% weight per waste horse genomic DNA used as the calibrant and diluted to a suitable working concentration as shown in the table. And finally, the actual thermal cycling conditions are standard thermal cycling conditions as shown in the table. So to summarise then, the draft Defra SOP for quantitation of horse DNA uses real-time PCR to quantitate the amount of amplifiable horse DNA relative to the total amount of amplifiable mammalian DNA present in a sample. EU and UK guidance is available regarding quantitation of meat species, and the 1% weight per weight raw meat value is a generally agreed and established level for enforcement action. This method has been developed and validated for use with DNA extracted from samples that consist of raw lean horse meat in a raw lean beef meat background, as explained earlier. Results can be equated to the equivalent amount of raw lean horse meat in a raw lean beef meat background on a gravimetric weight per weight basis. The method, however, has not been validated for all cooked and processed food samples. It will still correctly measure the DNA content, but may not give accurate meat content estimates in all cooked and processed food samples, similar to any other DNA approach.
I'd like to welcome everyone to this e-seminar presentation, which is part of the application of real-time PCR for food authenticity testing, inclusive of the quantitation of equine DNA e-seminar series. Today I will be talking about the draft SOP for the quantitation of horse DNA relative to mammalian DNA in raw meat samples, specifically the data analysis methodology, and provide worked examples. My name is Gavin Nixon and I'm part of the food authenticity team within LGC. So today I'll be talking about the SOP and its specific application to testing. So as part of the presentation, I'll be going through the SOP itself, focusing on the analysis methodology, show a worked example, and then provide a summary at the end. So you can see on this slide there's a decision tree. We prepared this and it's available through the SOP, basically to give users a kind of simplified decision process to follow when they actually conduct the analytical test. This are broken down into two parts. Part 1, which is shown on this slide, where essentially you've got the extracted DNA, calibrant materials, going through to the real-time PCR testing, and then there's various different decision points, leading from whether you've got amplification detected in your extraction control, or amplification detected in your NTCs, and then the kind of suggestions on how to proceed from these decision points. In part 2, this is kind of moving through the decision tree. You can see we go into the processing data, a certain metric parameters met, yes or no, carrying on through. This is just kind of ensuring the confidence in the data that you generate at the end. So just going on to the workflow itself. Essentially the first part is collating the qPCR data that you've generated using the SOP. Constructing calibration curves, which is essentially taking the log transformed copy number, which you've estimated based on spectrophotometric measurements, and then plotting that against the CQ, which is the threshold cycle threshold measurements. So to perform that for both assays, the couple, which is the horse specific assay, and the mammalian universal, which is the lab assay. Then fitting independent simple linear regression curves to that data set, deriving the performance metrics such as R squared, PCR efficiency. So you've got the equation of the straight line, which is used to calculate the coefficient determination, PCR efficiency, and then obviously using the performance metrics to evaluate the method performance, such as the R squared should be greater than 0.98. PCR efficiency should be within a kind of standard working range of 100% plus or minus 15%. And then using that data set to generate the kind of genome equivalent calculations. And then from that you can calculate the relative quantitative value and then report accordingly. So in this slide you can see the tabulated and summarized data sets specific to the horse and mammalian assays. We've got the standards presented as standard 1 to standard 7, the test sample itself, and then the log transformed calibrant taken from copy number to log 10 transformed number. Right. Now I'll be talking about the calibration curve metrics derived from the linear equation of a straight line, which is essentially y equals mx plus c, wherein y equals cq, or otherwise known as CT, which is dependent on which particular qPCR instrument you use, m, which is the slope or the gradient, x, which is the log 10 transformed copy number, and c, which is the intercept on the y-axis. You can use the calibration curve metrics to calculate PCR efficiency, which uses the slope. There's obviously the coefficient determination R squared as well, and the actual calculation for PCR efficiency is derived from using the slope and the equation specified in the slide. And then you use these metrics to determine whether they fall within the specified range, wherein the R squared is greater than 0.98, and the PCR efficiency is within a working range of 100% plus or minus 15%. Assuming that everything does, then you're fine to proceed on to the next step. So generating the calibration curves, very simple, essentially taking the equation of the straight line, using that to calculate the slope, intercept, R squared values, and then from those the PCR efficiency for both the horse specific and universal assays. Then using that to calculate your estimated copy number of your test samples. So you rearrange the equation of a straight line, which is, as you've seen previously, is y equals mx plus c. Rearrange that as stated on the slide, whereby the x is, as before, the log 10 transformed copy number, and the x asterisk is the estimated copy number once it's been transformed. So calculating the relative quantitation value. So you calculate the estimated relative quantitative value of the horse DNA compared to total mammalian DNA using the following equation, wherein the calibration curve derived horse genome equivalent copy number is divided by the calibration curve derived mammalian genome equivalent copy number multiplied by 100, and that gives the percentage horse meat DNA content within that test sample. The value provides an estimate of the horse content of a sample on a gravimetric weight for weight basis, provided that the sample is comprised of raw horse meat in a raw beef meat background. So now I'll move on to a worked example, just to give you a feel for the application of the SOP analytical methodology to hopefully a kind of a real life sample. So as part of the worked example, the initial stage is the generation of the test materials. So that's essentially extracting the DNA from the calibrants and test materials. So that would be your meat sample and your horse, 100% horse material. Then evaluating that extracted DNA quality, because that's obviously very important for qPCR based analysis, in which the A260 to 80 measurement is found to be around about 1.8, and the A260 to 30 ratio is around about 1.8 to 2.2. So these are spectrophotometric derived measurements. Obviously, if you're not using a spectrophotometer and you're using something like a pico green based measurement, you won't receive these quality indicators, which is why I would always recommend that you use a spectrophotometric based testing approach. There's also kind of various different guidance processes available. There's a Defra DNA extraction KT event, which was delivered in March last year. That's 2014, and that will be available through the Food Authenticity Network at a later date. In addition, there's also the decision tree, which you can see a scaled down version on this slide, which is also available through the SOP itself, and that should help analysts guide their testing process. So performing the actual real-time PCR analysis on the test materials, this slide shows kind of idealized results. So you can see the amplification plot here, amplification curves, and for both assays. They indicate good assay performance, nice good linearity shown with the calibration curve. You're getting nice delta RNs with regards to the peak fluorescence for the amplification plots, and you get a nice R squared as well. So that indicates that the test system has performed as expected. So you've initially got a preliminary evaluation of assay performance, so looking for performance characteristics such as good and consistent amplification curve, which both of these are showing, good baseline, no artifacts, good delta Rn, which is basically the difference between the baseline measurement and the maximal fluorescence of your test sample, no amplification in the NTC, which is obviously very important. The amplification in the extraction blanks, again, equally important. And then taking those data sets and exporting the CQ values for further analysis in a software package such as Microsoft Excel. So here are, in this slide, we're summarizing the data sets derived from that PCR run, qPCR run. As you can see, we've got the standards labelled 1 to 7, the test sample for the horse specific and the mammalian specific assays. Then we have the calibrant quantities that are being transformed into the log 10 quantities. And then we can use that data set to generate calibration curves for the horse and the universal mammalian assays, as you can see on this slide, which involves plotting the mean CQ value against the log 10 genomic copy number, fitting a simple linear regression curve to each calibrant data set, and then using that to derive the performance metrics using the equation of a straight line, which is y equals mx plus c, and then which is helpfully available through software packages such as Microsoft Excel. And then from that you can output the slope, which is m, intercept c, and get information on r² as well, and also it allows you to calculate PCR efficiencies, which, as you can see from this particular example, the r² are tight, they're all above 0.98 for both assays, and the PCR efficiencies are all above 90% as well, which falls within our recommended specifications. So performing the copy number calculations is a simple process. So essentially you take the estimated copy number calculated from the CQ minus the intercept over the slope, and then use that to work out the estimated copy numbers. So using the figures that we've put into the slides there for the horse copy numbers, which for this particular test sample came up to 80 copies. So once we've calculated that, we would do the same for the mammalian copy number as well. Calculate that. As you can see, that's a much higher number as you would expect, and because that is the total mammalian DNA background. So then performing the relative quantitative analysis is calculating percentage horse to mammalian DNA ratio, which is taking the horse genomic copy equivalence, HC, over the mammalian genomic copy equivalence, MC, and then multiplying it by 100, which in this particular test scenario is the estimated horse genomic equivalence of 79.97, the estimated mammalian, which was calculated to be 7963.01, and then from that we can derive the estimated horse adulteration content, and very conveniently that comes up to about 1%. In summary, the e-seminar has dealt with the application of the draft standard operating procedure for the quantitation of horse DNA relative to mammalian DNA in raw meat samples to a simple worked example. Looked at the importance of guidance tools and performance metrics. Discussed performance metrics such as R² and PCR efficiency, and highlighted how important they are to monitor during the assay process. The method itself can be applied to the measurement of amplifiable horse DNA present within any sample. However, it is currently only validated for use with DNA extracted from samples that consist of raw lean horse meat in a raw lean beef background. I would like to thank everyone for the attention, and for further information please visit the Food Authenticity Network website where you can find other resources.
I'd like to welcome people back to this e-seminar on the application of real-time PCR for food authenticity testing, inclusive of the quantitation of equine DNA, and we're going to briefly go through some aspects related to methods for food authenticity testing and availability of reference materials. So, two of the priority areas identified as part of the knowledge transfer event on the 17th of September 2015 with respect to real-time PCR for food authenticity testing was the availability of methods for such testing and also the availability of reference materials to provide confidence in the results of the food authenticity testing methods, and these are the two subjects we're going to briefly touch upon now. So, example methods for food authenticity testing. Examples of pre-existing food authenticity testing protocols are provided on the screen here, including pasta, looking at durum wheat and the adulteration of durum wheat, basmati rice, fish speciation, GMOs, meat speciation, etc. Now, these scientific reports and protocols were formerly available through the Food Standards Agency Foodbase repository, which is now archived at the National Archives. What I'd like to do is introduce the Food Authenticity Network website, the web address you can see at the bottom of the screen there. And LGC was appointed the coordinator for the UK's Virtual Food Authenticity Network, which is a Defra initiative earlier this year in 2015. The purpose of this Food Authenticity Network is to raise awareness of tools available for stakeholders to check for mislabelling and food fraud, but equally well to ensure that the UK has a resilient network of laboratories for food authenticity testing in general. And the Food Authenticity Network is overseen by Defra's Advisory Steering Group and supported by the RSC. Again, the aim of the network is to provide a web-based tool for food authenticity testing, objectives being to provide a one-stop shop for this, also to provide a forum for sharing information and best practice, and to facilitate efficient dissemination of information. The full website itself will provide opportunities for dissemination and networking of knowledge, information from previous and current knowledge transfer events, and also the ability to host e-seminars. And the pilot web-based tool went live on the 14th of July 2015. So in relation to the current e-seminar, the library for food authenticity related documents is currently under development on the Food Authenticity Network, but this provides links to research reports, authenticity surveys and standard operating procedures which may be of use to the listeners. So the way to access this is to go through to the Food Authenticity Network website itself, or click on Research at the main menu as indicated here. This will bring you through to a new web page and will include reports from the previous FA01 Food Authenticity Programme, as well as reports and SOPs from the Q01 projects which are funded under the Food Authenticity Programme. Just to highlight a few of these reports and protocols that may be of interest to listeners here. I've got some example methods and protocols available for food authenticity testing. From FA01 we can highlight such things as a SNP approach for cattle and pig breeds in project FA0112, collaborative trial for basmati rice in FA0110, a very good report on quantitation of horse DNA, FA0135, which I would encourage all our listeners to have a look at. Equally well, there are some protocols and project reports from Q01, Food Authenticity Programme, including approaches for looking at authenticity of fruit juice, fish, meat, Durham wheat as a real-time PCR approach, and also exotic meats inclusive of bushmeat. Here we've just summarised some of the example DNA approaches with the references that are available, the project references, as well as what approaches were used for the food authenticity testing. For example, PCR, restriction fragment length polymorphisms associated with PCR, real-time PCR, microsatellites, etc. So meat speciation is often a problematic area. There's a number of approaches and aspects associated with this that need to be considered. For example, looking at generic markers, perhaps on the mitochondrial genome, versus specific markers. These can allow you to look at differences between species. For example, is the meat pork or is it beef in origin? As well as looking at varietal differences. For example, is this beef Aberdeen Angus? There's many published papers on the subject, which you may like to avail yourselves of. Many of the earlier published papers do suffer from issues of cross-reactivity, so that's something to be aware of. Best advice we can provide on this is again to look at protocols available on the Food Authenticity Network, or to contact ourselves at LGC or the Food Standards Agency or Defra. There are a number of approaches available. Many revolve around using real-time PCR, and in some instances the use of the adjuvant bioanalyzer. And there's many methodologies available. Of course, there's the LGC Defra SOP, which we've described earlier, as well as off-the-shelf kits, for example, one provided on the screen there. Now, the best advice we can give for commercially available kits, there's a plethora of these available. It's often difficult to choose specific ones. The advice we can provide really is to speak with your colleagues on this via word of mouth to see what kits are appropriate. Also to check within the kit if validation data is available. If not, you can often contact the manufacturer of the kit itself and ask for validation data to be provided. And validation data will provide you with information on the assessment of important performance characteristics that you may need to consider for your particular application, including the lower limit of applicability, or the LOD, the limit of quantitation, how selective the assay is, as well as the accuracy inclusive of trueness and precision estimates. So moving on to the second part of this presentation, which is to provide advice on example reference materials for food authenticity. There are multiple sources of reference materials available. We've listed 3 on the screen here. Reference materials can be provided and sourced through LGC Standards, which is an authorised producer and distributor of reference materials with the appropriate accreditation, as shown on the screen. Other avenues to explore for reference materials include, for example, the American Oil Chemists Society, and also the European Commission Joint Research Centre and the Institute for Reference Materials and Measurements, which is part of the JRC. LGC Standards produce reference materials, and again there is a website at the bottom of the screen that you can go through to avail yourself of what reference materials can be provided and supported through standards. An example is shown on the screen here. A number of reference materials are available, including ones for food and beverages, inclusive of reference materials for allergens, animal food products, cereals and GMOs, fish and fish products, etc. Again, there is a link at the bottom of the screen that I would encourage listeners to make use of to get through to the appropriate website there. Aside from LGC standards, there are of course other avenues to explore for reference materials. One example here is the American Oil Chemists Society, or AOCS. Again, there is a link at the bottom of the screen there. They provide a range of reference materials, inclusive of certified reference materials for a range of genetically modified organisms. And finally, as mentioned earlier, there's the European Commission, JRC, the Joint Research Centre, which provides a range of reference materials for a number of applications, including clinical and environmental applications, food analysis, which may be more important here, fuel analysis, looking at GMOs, etc. We've got an example of the range of GMO reference materials that are provided by the Joint Research Centre on this screen. And the next screen shows a typical certificate associated with certified reference materials provided by IRMM, part of the JRC. And this certificate is associated with a certified reference material for GMOs, which I believe is a Roundup Ready soya example. And the certified reference material with the certificate shows a couple of values here, which shows that the certified value is 10% plus or minus 0.7% as the uncertainty value associated with the material. Just a note about certified reference materials. The best reference standards, certainly for GMO analysis and equally for any analysis, would be the use of certified reference materials. For GMOs, these are provided by IRMM and distributed through LGC standards. These are reference materials that have been certified for their GM content, for example based on a mass per mass approach, which is the most common approach, but it also can be certified on the ratio of a transgene to endogenous target on a smaller range of plasmid certified reference materials. Again, I would encourage listeners to use certified reference materials where possible, as all CRMs are accompanied by a certificate detailing the measurement uncertainty associated with the reference value, which can then be taken into account as part of the analysis any lab will be doing to estimate their own uncertainty budget. So to summarise there are a number of validated protocols available for food authenticity testing. I would encourage people to take a look at the Food Authenticity Network, as shown on the screen, which is acting as a repository for a number of reports and protocols available. There are various commercially available kits for food authenticity testing. Again, I would encourage people to demand to have a look at validation data and to look at the performance characteristics enclosed therein to make a qualified judgment as to whether that kit is fit for purpose for their own application. And finally, there's a note on reference materials. We've got 3 examples of providers of reference materials here, including LGC standards, IRMM as part of the Joint Research Centre, and AOCS. And again, where possible, always insist on choosing certified reference materials. I'd like to welcome people back to this last presentation regarding an e-seminar on the application of real-time PCR for food authenticity testing, inclusive of the quantitation of equine DNA, whereupon I will provide a presentation just to summarise some of the prior events. So this e-seminar aim was to provide access to information presented at the original joint Defra Government Chemist Knowledge Transfer event held on the 17th September 2015 at LGC regarding the application of real-time PCR for food authenticity testing, inclusive of the quantitation of equine DNA. This was a dual knowledge transfer event focusing on 2 distinct but related topics, the first of which was the application of the LGC Defra draft SOP for quantitation of horse DNA, and the second aspect was to look at the application of PCR and real-time PCR to food authenticity testing in general. So to summarise the seminar and the pertinent points at the knowledge transfer exercise itself, we talked about a real-time PCR approach for quantitation of horse DNA, the background behind the work and the SOP itself, the need for such an approach. We touched upon some of the prior Defra projects on the development and method validation of real-time PCR approach. Also the draft standard operating procedure, the purpose behind that, its scope and criteria, the application of the SOP, the principles behind that method, and also the evaluation of some of the important performance characteristics associated. We then went on to talk about some of the data analysis and provided a worked example associated with the application of this SOP. And finally, we rounded off with some of the food authenticity testing methods and reference materials, including talking about the availability of validated protocols through the Food Authenticity Network website, some of the commercially available kits, and some advice on criteria on selection of these kits. And the last point was the availability of reference materials, and we provided 3 examples of sources of reference materials, including LGC standards, AOCS and IRMM as part of the Joint Research Centre. And where possible, again, the advice was to source appropriate certified reference materials. That brings us to the end of this series of presentations in relation to this e-seminar. I'd like to thank everybody for their attention and listening to myself and my colleague Gavin Nixon for the presentations of these e-seminars. We can be contacted on the details below, and we are looking forward to providing some additional e-seminars associated with other previous knowledge transfer events, including real-time PCR analysis, DNA sequencing and DNA extraction work which was done as previous Defra KT events. Thank you.